Good morning, my dear students of class 11 science. Welcome to biology class. So we'll continue with the chapter anatomy of flowering plants. In the last class, we have learned about the anatomy of roots, that is dicot root and the monocot roots. So in today's class, we'll continue with the topic internal structure of dicot and monocot stems. Okay, so we are going to learn about the anatomy of dicot stem and the monocot stems. So first of all, we'll learn about the anatomy of dicot stem. Okay, dicotyledonous stems. So mainly in the transverse section, remember that TS means transverse section. Okay, LS means longitudinal section and TS means transverse section. So on a transverse section, the young dicot stems will show the following structures. So what are those structures? First of all, we'll see the presence of epidermis, then we'll find the cortex. Okay, we'll come back into hypodermis when we'll go through the cortex part in detail. Then we'll see the presence of pericycle. Okay, so here again you can see the presence of endodermis also. So these three part, hypodermis part, cortex part, endodermis part, we'll learn in detail when we'll go through the structure of the dicot stem in detail. Okay, so we'll find epidermis, we'll find cortex, we'll find the presence of pericycle, we'll see the presence of vascular bundle, that is xylem, phloem. Okay, then we'll see the presence of medullary ray or the pith ray, and finally we'll find the presence of pith or medulla. So this is just the transverse section of our dicot stem. Okay, so now we'll go in detail and we'll learn about the internal structure of the dicot stem. Okay, so first of all, the first part is epidermis, which is the outermost layer of the stem, and it is protective in nature. Okay, now this particular epidermis, it is covered by a thin layer of cuticle. Even you can see over here. So this particular part with no intercellular space, it is the epidermis part. Just above the epidermis, you will find a thin layer over here, which is called the cuticle. Okay, so this epidermis is covered by a thin layer of cuticles, and this epidermis, it also bears the trichomes and a few stomata. Clear? Now, after epidermis, we will find the presence of next layer, which is called the cortex. Okay, but remember carefully that cortex is divided into three subzones. Okay, so cortex is divided into three subzones. Those three subzones are hypodermis, cortical or general cortex, and the endodermis. These are the three subzones of the cortex layer. So that means this particular layer of cells is called the cortex. Okay, and it divided into three subzones: hypodermis, cortical or general cortex, and the endodermis. So cortex are generally cells which are arranged in multiple layers between the epidermis and the pericycle. So here you can see the presence of pericycle, and here we have got the epidermis. So in between the epidermis and in between the pericycle, we will find the cortex part. Clear? Okay, now I've told you that we have got three subzones of cortex. So first one, we'll talk about the hypodermis part. Okay, so you can see over here the hypodermis, the first subzones of the cortex, which is nearly 4 to 5 cell thick. It is in the page number 6.28. One more time, it's in the page number 6.28. Okay, so hypodermis is nearly 4 to 5 cell thick layer, and is made up of collenchyma. Okay, it's made up of collenchyma, and it just lies below the epidermis. So when we see the internal structure of the dicot stem, so just below the epidermis, so here you can see the presence of epidermis. So just below the epidermis, we will see the presence of hypodermis, which is generally 4 to 5 cell layer thick. Okay, and it is made up of collenchyma cells, and these collenchyma cells are the living cells, and it contains the chloroplast. You know what is important, the chloroplast. Okay, so it contains the chloroplast, and the function of this hypodermis is to provide mechanical strength and support to the young stem. Okay, now let's go to the second subzone, which is called the cortical or the general cortex. So the second subzone of cortex is general cortex or cortical, and it just lies below the hypodermis, and it is made up of parenchyma cells. Remember this thing, hypodermis is made up of collenchyma cells, but this general cortex or the collenchyma is made up of parenchyma cells. And when we look into general cortex, you will see the presence of intercellular spaces. So over here, this was the hypodermis portion, this was the hypodermis portion, and just below the hypodermis we can see the presence of this portion. Now okay, we can see the presence of general cortex. So in between the cells we can see the presence of intercellular spaces. So the cells of the general cortex has got intercellular spaces, and it is reduced to 1 to 2 layer outside the vascular bundles also. Okay, now the function of this general cortex or the cortical is to store the food and other materials and to provide the rigidity, the stiffness to the stem. Now the third layer or the third subzone of the cortex is the endodermis. It is just a single layer of cells. Okay, it is a single layer of cells, and it is the innermost layer of the cortex. Okay, and when we look into the endodermis, we can see there is no intercellular spaces, and it is made up of barrel type of cells with no intercellular spaces. Okay, and this endodermis used to store the starch, so it's also called the starch sheath. So simply, cortex just lies below the epidermis, and it is divided into 3 subzones: hypodermis, which is made up of collenchyma cells. There is no intercellular spaces. Then we have got the general cortex or the cortical cells, which is made up of parenchyma cells, where we can see the presence of intercellular spaces. They also provide rigidity to the stem cells and stores food and other materials. And the innermost layer of the cortex is the endodermis, which is mainly a single layer of cells. We don't find intercellular spaces over there. It is made up of barrel type of cells. Okay, and functions is that it stores the starch grain, so it is also called the starch sheath. So these are the 3 subzones of the cortex. Now just below the cortex we will find the next portion, which is called the pericycle. Okay, so now we are going to learn about the pericycle portion of the dicot stem. So here you can see the pericycle. So this is also the pericycle part. Okay, and even this is the pericycle part. Alright, so you might be confused. So now you can see over here. So this one was the epidermis part. Okay, this one is the hypodermis part. Then we have finished cortex, and inner layer is the endodermis. Now we are going to learn about this structure. This is the pericycle part, and you can see it is present throughout the dicot stem. Okay, so let's check it out. So pericycles, it is formed of sclerenchyma. Listen very carefully. It is made up of sclerenchyma above the phloem. Okay, and it is nearly 4 to 5 layer thick. The cells of this pericycle, that is sclerenchyma, they are the dead cells and rigid. Okay, so the sclerenchyma cells that makes the pericycles are dead cells. They are rigid and with their walls considerably thickened and lignified. Okay, so the function of pericycle is that it provides mechanical support to the plant and protect the vascular bundles. So the function of pericycle is to protect the plants and the vascular bundles, and it also stores the food. Okay, now next one we'll talk about the vascular bundle. Okay, so vascular bundles over here, it says that it is conjoint, okay, collateral, open with enlarged Proto Xylem. Now what does conjoint means? It's very important, okay, because we are saying that vascular bundles are conjoint, collateral, open with enlarged Proto Xylem. What does conjoint means? So when xylem and phloem combined in the same bundle, very carefully, when xylem and phloem, when they combine in the same bundle and when they are present on the same radius, then it is called conjoint. Now what does collateral means? Collateral means xylem is toward the inner side and phloem on the outer side. That is called collateral. So xylem is present toward the inner side and phloem is present towards the outer side. That is called collateral. Open means when we can see the presence of cambium between the xylem and the phloem. Okay, so that is called open. So look over here. So this one is the xylem. Okay, this is the center part, which is called the pith. So xylem is present toward the inner side. Okay, and you can see the phloem. This is the phloem part. Okay, phloem is present towards the outer side. So when xylem is present toward the inner side and the phloem toward the outer side, it is called collateral type. Clear? And open means we have told you that there is the presence of cambium in between the xylem and the phloem. So this innermost part, okay, this innermost part is called cambium and is present between the xylem and the phloem. So it is called open type. Now the next term is enlarged. What does enlarged protoxylem mean? If you remember, we have learnt about the exarch cases where protoxylem was present outside and metaxylem was present inner side. But here in case of enlarged protoxylem, the protoxylem will be present towards the inner side and the metaxylem will be towards the periphery or towards the outer side. So it is called enlarged protoxylem. It's just the opposite of the exarch vascular bundle. Okay, so we have learned that the vascular bundles is conjoint, collateral, open, and they have got enlarged protoxylem. Now in between the vascular bundles we will see the presence of few parenchyma cells also. Okay, now vascular bundles is mainly composed of phloem, xylem and cambium. So even I have shown you that figure also, figure number 6.26, where I have shown you the structure of the xylem, phloem and the cambium. So the xylem, phloem and the cambium, it will form the vascular bundle. Okay, so let us talk about the phloem. Okay, so when we look into the structure, figure number 6.26, I have told you which one is phloem. So this one is the phloem part, the inner one is the cambium, and this is the xylem part. Okay, so phloem lies toward the outer side. Very carefully, phloem lies toward the outer sides and is mainly composed of sheath tubes, companion cells, and the phloem parenchyma. This is the composition of phloem. Okay, so this phloem is present towards the outer side, and this phloem is made up of sheath tubes, companion cells, and the phloem parenchyma cells. Okay, the cells of the phloem are living cells and they have a thin cellulose wall. Then let's go to xylem, which is the innermost layer of the vascular bundle, because xylem is present towards the inner side. This is the xylem. Okay, so it is present towards the inner sides. Okay, towards the center of the stem, that is pith. Now xylem is mainly composed of basials, tracheids, hood fiber and hood parenchyma. Now the small vessels, let's see the structure. If you look over here, that xylem is mainly divided into protoxylem and metaxylem. So don't get confused. So we have got metaxylem and we have got protoxylem. These are the types of xylem. So the smaller vessels, if you look at the structure of the xylem, and you can see that the smaller vessels mean the small structures. Okay, so this bigger one is the metaxylem, and so this is the metaxylem, and the smaller vessels, these are the protoxylem. So the smaller vessels which lies toward the center, which coming toward the pith. Pith means center of the stem. Okay, the smaller vessels which lies toward the pith, toward the center of the stem, it forms the protoxylem, and the bigger one that moves away from the center, that moves away from the pith, is called the metaxylem. So such a type of xylem is called the enlarged xylem, because what enlarged means, protoxylem lies towards the center, lies towards the pith, whereas metaxylem lies towards the outside or lies in the periphery. So this type of arrangement is called enlarged. Had it been that the metaxylem was towards the center and protoxylem outside, we will call exarch. But here we are finding the enlarged type of vascular bundles because protoxylem is facing towards the pith, towards the center, and metaxylem away from the center. Next one is cambium. This is present in between the xylem and the phloem, and shown in the diagram also. The cambium cells are small, they are rectangular, they are thin-walled, and they are radially arranged. So in between the xylem and the phloem, we'll see the presence of cambium. So this part is the xylem, metaxylem, and the protoxylem. This one is the phloem. So in between the phloem and between the xylem, we'll see the presence of cambium. So this is the cambium. Okay, so this type of arrangement is called open type of vascular bundles. Now next one is medullary ray. So we're going to talk about the medullary ray or the pith ray, so which constitute the radial root of the big rectangular parenchymal cells, and it is present in between the vascular bundles. So this is the first vascular, let's say suppose this is the first vascular bundle, and suppose this is the next vascular bundle. So in between the two vascular bundle, we will see the presence of this one. We will see the presence of medullary ray or the pith ray. Okay, so lies in between the vascular bundles, and they serve in the conduction of water and food in the radial direction. So when we do the structure, so here in suppose this is the first vascular bundle, this is the second vascular bundle. So in between these two vascular bundle, we will see the presence of medullary ray or the pith ray. So here also in between the two vascular bundle, we will see the presence of medullary ray or the pith ray. Clear? Now the next part is the pith or medulla, which is the central parenchymal zone, and in case of sunflower, it will occupy the major portion of the stem. The function of it is that these parenchymal cells are the living cells, okay, and these particular cells, they are thin-walled and they are rectangular with conspicuous intercellular spaces. So pith or medulla is the center of the stems, and here it is made up of parenchyma cells. So here we have finished the structure of the internal part of the dicot stem. Now we will see the internal structure of the monocot stem. TS means transverse section. So when we go into the transverse section of the monocot stem like the image, we will see the presence of epidermis, we'll see the presence of hypodermis, we'll find the ground tissue, and we'll see the presence of vascular bundles. Just look at the structures very carefully. You can differentiate the structure of the dicot and the monocot stem. In dicot, we were learning the structures like pericycle. If you remember that we are finding structure like pericycle, all those things were there. But here we are not finding structure called pericycle. We are not finding structure called endodermis. Okay, we are just simply seeing the epidermis, hypodermis, ground tissue, and vascular bundle. So let's go in detail. First one, epidermis part, the outermost layer of the stem, and which provide protections. And also cuticularized, it contains the cuticle. We find thin layer of cuticles of the epidermis. Okay. Now, just below the epidermis, we will find the presence of next layer, which is called the hypodermis over here, and is made up of sclerenchyma cells. It just lies below the epidermis, remember. Okay, and after that is made up of 2 to 3 layers of sclerenchyma cells. So when we look over here, just below the epidermis, 2 layer thick, we can say, is the hypodermis, and it is made up of what type of cells? It is made up of sclerenchyma cells. It also provides mechanical support and strength to the stem. Then comes the next one, which is called the ground tissue. Okay, the ground tissue. Now, like in case of the dicot stem, in monocot stem, this ground tissue is not differentiated into definite tissues like cortex, endodermis, and pericycle. These cortex, endodermis, pericycles can be seen in the dicot stem, but here in monocot stem, the ground tissue is not differentiated to form the cortex, to form the pericycle, and to form the endodermis. Okay, and when we look into the structure of the cells of the ground tissue, we can see the presence of intercellular spaces. Okay, and this ground tissue is made up of parenchyma cells, and it extends from the hypodermis to the center of the stem, that is the pith. Okay, next one, we'll talk about the vascular bundle. It's very important. So here also you can see that vascular bundles are collateral, they are conjoined, and they are closed. Okay, That means here we don't see the presence of the cambiums, all the cells. So vascular bundles are collaterals. I've told you already the meaning of those things, what does collateral means, what does conjoint means, and what does closed means. Okay, and open type also we have discussed. Now here it said that it is surrounded by a bundle sheath. It provides protections and strength to the vascular bundle. Clear? So first thing you remember here that vascular bundles, they are collateral, they are conjoint, and they are closed. Now here you get two terms, open and closed vascular bundles. When we were discussing about the Dicot stem, we have seen the presence of cambium between the xylem and the phloem. That first diagram, if you remember, we can see the presence of cambium in between the xylem and phloem in structures, right? So it was called open type. But here we don't see the presence of cambium in between the xylem and the phloem, so it is called closed vascular bundle. Clear? Okay. Now, vascular bundles, they are surrounded by a sheath of thick cells called sclerenchyma cells. That's why it is also called the bundle sheath. Okay. Now we'll just see the structures of the vascular bundle, that is xylem and phloem. So first of all, let's talk about the xylem. Clear? So I am just going to magnify the structure of a single vascular bundle, that means this one. So these are the different types of vascular bundle. We can say this is the first vascular bundle. Okay, we have got the second vascular bundle. We have got the third vascular bundle over here. We have got the fourth one also, right? We have got the fifth one also over here. So now we are just going to look into the structure of one vascular bundles, and we are going to discuss about the xylem and the phloem. Okay, so let's see. So this is the magnified view of a single vascular bundle. So let's talk about xylem. So xylem consists of four distinct vessels which are arranged in the form of Y. You can see over here these structures, four distinct vessels, 1, 2, 3, and 4. So it looks like structure of a Y. Okay. Now there are two larger vessels, this one and this one. These are two larger vessels which is called the metaxylem, and there are two smaller vessels, these two, which are called the proto xylem. Okay, so two large vessels are called metaxylem and two small vessels are called proto xylem. Clear? Okay. Now next one comes the phloem one. The phloem lies in between the arms of Y facing the epidermis and consists of sheath tubes and companion cell. That means which part is phloem 1? It said that it lies in between the arms of Y facing the epidermis. This upper part will see the presence of epidermis. So here, this is the xylem. These two are meta and these two are proto xylem. So this one is the phloem. Okay, so this one. So this one is the phloem, which is facing towards the epidermis and is mainly composed of what type of tube? Sheath tubes, and we can see the presence of companion cells. So it is facing towards the epidermis and it lies just between the arms of the metaxylem. Okay. Phloem parenchyma is absent. We can see the presence of companion cells, we can see the presence of sheath tubes, but phloem parenchyma is absent in case of monocot stems. Now the protofluent, which is broken mass of the cells, right, which lies on the outer side and the inner portion, it forms the metaphlloem. So it's just the structure of a single vascular bundles where you can see the presence of the xylem and the phloem. Xylem with four distinct vessels which are like forming a Y structures. Two larger ones are called the meta and the two smaller ones are called the proto. Then phloem lies in between the arms of the Y facing the epidermis and is mainly composed of sheath tubes and the companion cells, and phloem parenchyma are absent. So here we have finished the internal anatomy or the structure of the dicot and the monocot stems. Now from your textbook, kindly go through the page number 6.31, go through difference between the dicot stem and the monocot stems. So this much for today. In the next class, we'll learn about the anatomy of leaves. So see you in next class. Thank you.